Welcome to PMART at Pacific Northwest National Laboratory. In this video, we will walk through an example use case of PMART cancer. There are two experiences to choose from. The first is still under development, and this is to upload a personal dataset. The second is to explore an existing dataset from CPTAC. We can choose to search datasets either by cancer type. Currently, there are two, ovarian and breast. More datasets are added as they become available. Or we can search datasets by experiment type and search for either protein, peptide, or gene datasets. For this video, we will select a subset of the ovarian peptide data before parsimony. On the left, we see a summary of the chosen dataset. We can see that there are 84 samples and just over 2,000 peptides. Also shown are the numbers of proteins and genes that those peptides map to and the percent of missing data. We must select a main effect variable that will be used for the statistical comparisons to be made. Once we've selected a variable, we must choose a grouping. For instance, clinical stage has one option for grouping. And we can see that there are 7 stage 1 to 2 tumors and 77 stage 3 to 4 tumors. We will select tumor residual disease and can choose to group the data into 2 groups of no macroscopic disease or 1 to 10 millimeters, or those tumors that were greater than 10 millimeters, or use the data grouping shown by selecting as is. In this case, we can see that there are 10 tumors that did not have a tumor residual disease given in the dataset. Finally, we have the option of selecting a covariate, which we can account for in our analysis, but for which we're not interested in the direct statistical comparisons. On this page, we select the modules to include in our workflow. We can select them individually, notice that when we pull in modules with dependencies, those dependencies are automatically added to the workflow. To remove modules, either click the X to remove individual modules, or we can clear the entire selection. A suggested workflow is also available. Additional modules can be added or subtracted. The first filter is a peptide filter, which retains peptides that meet a minimum peptides per protein criteria. Clicking Apply Filter shows a preview of the number of peptides, proteins, and genes, both before and after the filter. Once we're happy with the filter selection, we click Next to apply the filter to the dataset and move on. This module shows a sample outlier filter, which identifies potential outliers based on several metrics. More details about this algorithm can be found in the reference provided. In this graph, each plot corresponds to a sample. Samples colored in red above the line are the potential outliers. By hovering over a point, you can see the values for the various metrics that were used. We can move the threshold for identifying outliers by either dragging the horizontal line up or down, or by manually altering the p-value threshold in the box and clicking somewhere on the page. Any samples that are above the line will be removed from analyses going forward once we click Next. The peptide coverage filter removes any peptides for which we are unable to perform a statistical test. There are two tests available: a qualitative test for missing data and a quantitative test. You can filter based on either or both of these tests. Applying the filter shows how many peptides, proteins, and genes will be removed. The coefficient of variation filter filters peptides based on the CV. We can see that for this data set, the median CV is 36.73, and a default threshold of 90% is shown in this graph as the shaded blue region. This indicates the number of peptides that would be retained by this filter, and the number that are outside of the shaded area indicates those that would be removed. To change the threshold, we can either move the slider bar left or right, or manually enter a value. At a threshold of 100%, we can see from the table and the graph that no peptides would be removed. In this case, we're fine with that, so we will continue. To normalize the data, we must select a normalization and a subset method. The normalization methods are the functions for calculating the normalization factors, and the subset methods describe the subset of peptides on which to compute those factors. If you are unsure which normalization method and subset to use, there's an algorithm called SPANS that can help make that decision. This algorithm evaluates multiple combinations of normalization and subset methods and describes the bias introduced to the dataset if you choose that particular method. In this case, we will median center the data and compute the medians based on all of the peptides in our dataset. There are two options for performing differential statistics. We can either make all pairwise comparisons based on the main effect grouping that we chose at the beginning of the workflow, or we can choose to compare to a single control group. If we compare to a control group, then we must select the group for comparison. We will choose no macroscopic disease. These graphs show the number of peptides significant for each comparison by the different tests that we chose to apply. The threshold for significance defaults to 0.05, but this can be modified by the user. The different bars shown in the graph can also be toggled on and off. All of the graphs and tables shown throughout the modules are available at the end of the workflow in a summary. However, if you would like to download an individual graph, you may do so in that module. Finally, it is important to note that the p-value thresholds selected here will be used in future visualizations to restrict the data set down to just those significant peptides. We will use median quantitation to quantitate from the peptide to the protein level. Different quantitation methods were available for selection when we chose our modules at the beginning. A similar analysis is done for the protein level data as was done for the peptide level data. Again, we can modify the p-value threshold if we so choose to be used for down selecting the proteins in future visualizations. A probabilistic principal components analysis is done, which does not require imputation of missing data. This is computed using those proteins that were significant at our previous p-value threshold. In this plot, a user can change the colors used to represent each group if they wish. It is also possible to toggle on and off the groups that are shown in the plot. This module allows visualization of the individual peptides or proteins using the interactive visualization tool called Trelliscope. We will begin with the peptide level data. So we can see that we are looking at the first of 28 peptides that were significant at the p-value threshold we indicated on the peptide statistics module. This particular peptide is shown at the bottom along with the gene and proteins that are associated with it. The plots give both quantitative and qualitative summaries for this particular peptide. We can scroll through the 28 different peptides using the next and previous buttons. In this case, using the subset data set that we chose, this is reasonable, but if we were using the full data set and had many more peptides to explore, we might want to sort and filter them using the table sort filter. This shows various metrics about each peptide, including the counts in each group, means, and p-values. For instance, we can choose to sort based on the p-value comparing the 11 to 20 millimeter tumors versus the no macroscopic disease group by selecting the arrow. This sorts the p-values from most significant to least. Now the peptides should be sorted in order from most significant to least. We can go to panel labels to add the p-value for this particular comparison to the space below the graphs. Now the p-value appears below. We can also go to table sort filter to filter by one of these metrics. We will filter based on a flag for the direction of the fold change for this particular comparison. Here we are filtering down to those peptides that were less abundant in the 11 to 20 millimeter tumors than in the known macroscopic disease group. And now we can see that there are two peptides. To remove a filter, we go back to table sort filter and click the X of the filter that we want to remove. Now we are back to our original 28 peptides. We can go back to table sort filter and search for a particular gene, peptide, or protein of interest. This brings up a single peptide associated with our gene of interest. You can save individual plots of peptides of interest by clicking on the disk icon. And once we're done exploring the peptide level data, we can click Next to move on to the protein level data. The same functionality is available in Trelliscope for protein level data as for peptide level data. In either case, there are links below each plot that give genes and proteins associated with the peptide or protein. Clicking on the genes will take you to gene cards where you can learn more about that particular gene. And clicking on a protein will take you to UniProt. After completing all of the workflow modules, a summary is available that contains all of the tables, plots, and threshold choices made throughout your workflow. From this page, you can also download the dataset archive, which includes the peptide level data after filtering and normalization, and the quantitated protein data. It also includes the results of the statistical analyses, p-values, fold changes, and the counts and means that you saw in Trelliscope. Each of these files is available as a CSV. Thank you for watching our video, and please check back for more updates as this tool is under continuous development.